Hello everyone, welcome to another session. In this session, we will be discussing on principal applications of Agarose Gel electrophoresis and SDS PAGE. Agarose Gel electrophoresis is a method of gel electrophoresis used to separate mixed population of macromolecules such as RNA, DNA or proteins in the matrix of agarose. Now, gel electrophoresis means phoresis refers to the movement, electro refers to the applied electric field. In other words, electrophoresis is movement of molecules under applied electric current. For this to happen, the molecule of our interest must have either a negative and positive charge, and we need two electrodes which are oppositely charged so that our charged molecules can go to the opposite electrode. Electrodes that are the molecules that are positively charged, are called cations, and they move towards a negative electrode called cathode. Ions that are negatively charged are called anions, and they move towards the electrode called anodes, because ions move to their opposite charged electrodes. So here what you are doing is, in electrophoresis, you are applying electric current and then you are allowing the molecule to move in opposite direction so that it moves from one charge. For example, if it is a negatively charged ion, it will move towards a positively charged electrode. If it is positively charged, it will move towards a negative electrode. So you are allowing them to move under applied electric field, so that later on you calculate or measure the distance traveled by that molecule with respect to the electrode that is present, or with respect to other molecules that are present in the system, and then you try to locate these molecules as well as maybe in later on you can also quantify them. This movement, the whole movement of the ions or the molecules, since it will be taking place in a medium of gel, it is called gel electrophoresis. And most common type of gel that is used for the molecular movement is agarose. So that's why this is called agarose gel electrophoresis. This agarose is actually a linear polymer. It is extracted from a seaweed, and this forms a gel matrix by hydrogen bonding when heated in a buffer and allowed to cool. So on heating it is liquefied, it is in the liquid state. On cooling it solidifies, rather it gels. So, and since it is neutral, it allows easy movement of the molecules. The hindrance or the resistance by the medium is very minimum. That's why agarose gel electrophoresis is used. So here it is used to separate the DNA fragments more commonly. Here the separation is based on the size of the molecule, DNA fragment. Larger DNA fragments will move slow, whereas smaller fragments will move faster. That means if you allow all the DNA fragments, a mixture of DNA fragments under applied electric field given voltage and for a given pH as well as for a given time, for a given time. For example, you are running for half an hour. You are allowing the molecules to run for half an hour. Running means you are allowing the molecules to move. So this is called as running. So what happens if you allow them to a specific time for half an hour, for example? From the beginning to half an hour, each fragment will move at a particular speed depending upon its size. It might sometimes also depend on the charge that is present and the medium you are using. That's why the agarose gel is used, because there is minimum resistance to the movement. So now you are left with the size and the charge. So when you allow the elements to move like this, what happens? Depending upon the size and the charge, they move at a different speed. Smaller ones will move faster, that means they are more close towards the next electrode, and larger ones move slower. So that means you are going to get a different levels, positions for each fragment based on its length. Here there is one more important thing. So the DNA will be negatively charged. So you are allowing this negatively charged DNA to move towards the positive electrode. And remember, cathodes are negatively charged electrodes, anodes are positively charged electrodes. So you are adding or you are inserting the DNA fragments in the negatively charged electrode, because these are also negatively charged DNA. What happens? Equal charges repel. So these DNA molecules will start moving towards the anode, which is a positively charged. Negative charged DNA moves towards the positively charged electrode. And this movement, this speed will be depending upon the length of the fragment that is used. So the rate of migration is always proportional to the size. Okay, so once it is run, then you have to visualize the movement which is done by ethidium bromide, so that you will know exactly where it has moved. Then illumination can be done by using ultraviolet rays. Okay, because of fluorescence, you can check the position of the DNA that is present. Here you can also set multiple samples of DNA, and you can use a DNA ladder. DNA ladder means it will have DNA fragments of known length. You already know the length of each DNA fragment, so that mixture will also be run simultaneously. Now, what is the advantage of this is that you can compare the known with the unknown. You have an unknown sample, you have a known sample, you are going to compare the fragment length, the length it has moved. Okay, just like what you have done in chromatography, where you go for the retention this one. Okay, so how long depending upon the solvent distance and then the chemical that is moved, you are going to get that RF value. So in the same way here too, you can actually compare the known fragments of DNA with the unknown fragments of DNA. You can identify the DNA fragment length. So this is the setup of Agarose Gel electrophoresis. So here you can see there is a gel tank, the Agarose Gel sheet. It is also connected to the power supply, negative and positive supplies. The positive electrode and the negative electrode. And the negative electrode, remember, the cathode, and the positive electrode is the anode. Okay, so this is how the the final setup is. You apply the electric current. You can see here the on loading, after loading. Okay, once you switch on the power supply, and the because of the difference in their charges, these fragments are going to move at a different length. You can see different wells. Actually, there are like you see here, there are different wells. These wells are punched in a gel sheet. You make a small wells, and after that what you do, you load the DNA samples of different, maybe unknown samples, as well as one ladder DNA sample. Then simultaneously, since you are supplying the current, simultaneously all will start moving, and this movement will be always dependent on the fragment length. Okay, so and then what you do, you can also compare with the ladder. This is the ladder sequence, so it will have all known fragments. Then you can compare it with the ladder to see what might be the length of the DNA fragment. Now this running will be usually in the gel. Later on you'll have to use other techniques, like you'll have to go for autoradiography. You'll have to go for blotting technique, suitable blotting technique. Then you can get it on a sheet. Then you can expose it to UV light. You can locate it if needed by directly removing from the DNA DNA gel. From the gel you can just punch it off, and then you can also analyze the DNA, the molecular weight of the DNA. That is also possible. Okay, And this agarose gel electrophoresis has a major use in separating the proteins DNA and RNA, particularly the DNA estimation of the size, and it can also be used for the analysis of PCR products, or like in molecular genetic diagnosis or genetic fingerprinting. So it helps in separation of restricted genomic DNA before southern analysis, or even RNA before northern analysis. So you first run it, then from there you blot it to a sheet, then it can be further analyzed. So this is one of the steps in DNA fingerprinting also, where you actually separate the fragments of DNA to get a pattern of DNA fragments, and then this will be compared with the the reference sheets, like if you want to compare the child and the parent, if you want, or if you are trying to identify the culprit, you have a sample, then you take a DNA from the culprit and then run both of them simultaneously, get a sheet. In that you can compare the DNA fragments, because next in one more chapter we might be discussing about the DNA fingerprinting technology, where we'll come across this VNTR and all. So where you can actually compare the DNA composition of a particular person with anyone you need it. Okay, simply by using this agarose gel electrophoresis technique, first you separate the DNA fragments and then you blot them, then you can analyze them. So this is one of the steps. So you can actually estimate the size of the DNA by using another, like restriction mapping of the cloned DNA. It is generally used to resolve the circular DNA with the different supercoiling topology, and to resolve the fragments that differ due to the RNA synthesis. In addition to providing excellent medium for fragment size analysis, it also allows the purification of the DNA fragments. Okay, since here the DNA fragment of a particular length is going to be together, you can separate it out. You can select a DNA fragment of your interest, and then it can also be used to analyze. The sequencing can also be done. There is next the one more step, that is like one more technique which is similar to our agarose gel electrophoresis, that is called as a SDS PAGE electrophoresis. Here SDS stands for sodium dodecyl sulfate, which is a detergent, and PAGE refers to polyacrylamide gel electrophoresis. Poly P, acrylamide A, gel electrophoresis GE. So commonly known as SDS PAGE. So this is a kind of electrophoresis used for proteins, because here they are separated based on the polypeptide chain length. Agarose gel electrophoresis is used for DNA and RNA, whereas SDS PAGE is specifically used for proteins. PAGE is polyacrylamide gel electrophoresis, which is common for protein. SDS PAGE is for denatured proteins. Now if you want to analyze a protein, or you are studying an enzyme, after this electrophoresis, if you want to see the activity of the enzyme, then you cannot use SDS PAGE. In that case, you are going to use a non-SDS PAGE, where SDS is not used. Directly you are going to use the PAGE only. But it is not as accurate as SDS PAGE in separation of the proteins. Whereas when you use SDS, what happens, as I said, this is a detergent. Okay, so sodium dodecyl sulfate. So this is a detergent, and it will denature the protein, so that you are going to get a linear chain of a polypeptide chain. Disulfide linkages and all are broken. You know, the proteins, they have their own structures in the sense functional structures. They may be primary structures, secondary, tertiary, quaternary structures. But when they are in their quaternary structure or a tertiary structure, separation is little difficult. That's why it is always better to go with the primary structure. So here in SDS PAGE, the proteins can be separated based on the fragment length, but the protein will be present in its primary structure, in the linear polypeptide chain. That is why you are using the SDS. And like agarose gel electrophoresis, here also the separation will be based on the fragment length, the linear length, okay, of polypeptide chain. And what happens here, like in agarose gel electrophoresis, you are actually adding polyacrylamide, preparing a gel. Poly, as the name itself says, it is acrylamide which is polymerized in the gel. You are also adding the SDS so that the protein is denatured and you get a linear chain. And then what you do, you are going to load them in the well, okay, with a suitable buffer. So here using a buffer is very important. And after treating with the SDS, usually the proteins will become negatively charged. Okay, but actually each protein will be either positively or negatively charged depending upon the pH of the medium. That's why they are amphoteric chemicals. Except in their isoelectric field, except in that, because they carry net no charge, whereas in all other pHs they carry either positive charge or a negative charge. Whereas because of the treatment with the SDS, they are going to be negatively charged. So you load them in the negative electrode area, okay, and then you allow them to run. So these fragments will move towards the positive electrode depending upon the fragment length. Okay, and then like this you can actually separate them. Once separation has taken place, you will be also using a tracker. Okay, tracker dye, so that you will know exactly where the solvent font has reached, and then all the other fragments will be behind that. So you need a tracker dye, and then you stain these proteins using a protein stain called as comacin blue, or sometimes nowadays even they use the silver stains. Most commonly used recent stain is a silver stain. So either comacin blue or by using silver staining techniques, you can get the bands, which is similar to the band which you get in agarose gel electrophoresis. And then later on you can identify the fragment length by using a reference. Okay, so the application of this SDS page is it is used in different fields of biotechnology. Okay, used to separate the HIV proteins during the HIV test. Purity of the protein is also identified in this process, and this is also used for peptide mapping. Okay, and used for measure the molecular weight of the molecules, because the fragment length is proportional to the molecular weight. The size of the protein is estimated by this process, and polypeptide composition is compared in this process. So that is all for today. Thank you, and these are my references.